All right, so now it's my pleasure to introduce today's speaker, Mike Dietrich, who is professor in the Department of History and Philosophy of Science at Pitt since 2017, coming from Dartmouth. And he's also our fearless leader. He's a chair of the department, and we're very grateful for his leadership. He's written extremely widely on the history and philosophy of biology, mostly evolutionary biology, but also molecular biology. And it will take quite a while to review all his research interests, so we'll just throw a few things out there. He has written on scientific controversies, on scientific outliers, so scientists who are the margins and of science and who can be making important contribution on model organisms, on the role of drift, on the role and significance of drift in scientific evolution, and on many other topics, both in history and philosophy of biology. I believe he has still 2 books forthcoming, but Mike might correct me. Maybe one of them is already actually in press. The first one is a biography of the evolutionary biologist Richard Goldsmith, called Richard Goldsmith's Heresies, with the University of Chicago Press. And the second one is a book about the history and philosophy of drift in evolutionary biology with Roberta Milstein and Rob Skippert, which is called Survival of the Luckiest. Are they still forthcoming?
Oh, yeah.
All right, so we're really looking forward to read these 2 books, and I think there's going to be further occasion to have you at the center when they are out. But today, Mike is going to be talking about the politics of embryology, Johannes Holtfreter's flight from Nazi Germany. So Mike will have about 50 minutes, 55 minutes, and then we'll have a 30 minutes Q&A. All right, Mike, the floor is yours.
Great, thank you very much. Welcome, everybody. Today is going to be a little bit more historical than philosophical, but I trust that you'll all be fine with that. I want to talk to you today about one of the best embryologists in the 20th century, definitely, I think, the best experimental embryologist, a man named Johannes Holtfreter. Johannes Holtfreter, we're not getting movement on the slides. There we go. Johannes Holtfreter was forced to leave Nazi Germany in the late 1930s. He went to Cambridge and then ended up at the University of Rochester. So you see a picture of him here on the left side of your screen that was published by the University of Rochester. And as we'll see, Holtfreter was also quite a little artist, and so he would always make these diagrams. So this is a picture of him contemplating various sort of embryonic fates. Holtfreter is a very interesting character, because if you start to look at all of the biologists who were forced to flee, he's one of the only scientists that fled as a political refugee. He didn't have to leave because he had Jewish ancestry, but because of some political reason. And up to this point, no one has been able to figure out exactly why he had to leave. And so part of the story here today is just me telling you that I figured that out, but also to sort of put him in a larger context of how we think about these things. So Holtfreter was part of one of the most famous embryology labs in the world. He was a student of Hans Spemann. So Spemann's the kind of very serious guy at the bottom of the journal cover here. Together with Hilde Preschold, who then became Hilde Mangold, what they did is they did a surgical transplantation experiment from one embryo to another, where they took that little bit of embryo that's colored black, it's called the dorsal blastopore lip, and they transplanted it to the backside of another embryo. When they did that, instead of getting a single embryo developing, they got 2 embryos developing. And so they hypothesized that that dorsal blastopore lip was in fact the element that would organize embryonic development. They called it the organizer. There was something about this region of tissue, they thought, that controlled the entire process of development. This sort of discovery and the subsequent work on it would win Spemann the Nobel Prize, one of the sort of only Nobel Prizes given for straight up embryological research. Holtfreter was a student in this laboratory. This is one of the diagrams of what he thought his graduate student life was like. Spemann is there driving the carriage. All of the animals that they're sort of transplanting things with are sort of locked on top. The staff is there behind the carriage, And then the graduate students are literally the horses pulling the carriage. And then you see the guy with the whip and the dogs nipping at their heels. Those are literally the postdocs and the lab manager whipping the graduate students on. And of course, each of the horses is making a snide and sarcastic comment about how horrible it is. And Spemann is saying, this is horrible. It must be going really well. So it was kind of characteristic of German laboratories, very hierarchical. You did exactly what the doctor professor said. Very little room for talking back or disagreeing with Spemann as an authority figure. Now, the Spemann network was quite extensive. So not only do you have these German students, but you had a huge number of people coming from around the world to study this technique. It literally was the cutting edge in experimental embryology in the pre-war period. Being able to make these kinds of transplantations was seen as an experimental intervention into the mechanisms of development. And the network was incredibly extensive. So the bigger project here is to actually understand what happens within this bigger network. What happens when people move away from Spemann, literally, physically, and intellectually. And you can see that there's lots of people going to different sort of contexts and different kinds of understandings. Holtfreter is right there, sort of in the dead center of the picture with the big red arrow next to him. He is one of the 5 Spemann students who's forced to immigrate and ends up going to the United States. And so the question becomes, what did that forced immigration do to him scientifically and personally? So when we're looking at exiles from the Spemann school, I tend to put them in 2 sort of major post-war contexts. One is concerning the diversification of developmental biology. So one of the things that we know is that the kinds of topics, the kinds of things that people were researching diversifies, becomes much broader in the post-war period than it was in the pre-war period. And so one of the questions is, did this migration, this forced migration, lend that or cause that diversity in research? There's also at the same time a shift away from an organismic or holistic view of embryos toward research on lower levels, at the cellular and subcellular level, and even the biochemical molecular level. And so there's this general move toward a more reductionistic perspective. And so the second related question is, did exiles, people who were forced to migrate, become more reductionistic as a result of that forced move? In other words, could this be a factor in explaining both the diversification and this move toward greater reductionist attitudes in developmental biology? On the one hand, there's sort of easy answers. Diversification, if you look at some people's cases, was obviously caused by their forced migration. Here's a very clear-cut case. This is Victor Hamburger. He was actually the lab director for Spemann's lab. He was also Holtfreter's best friend. Victor Hamburger, the whole Spemann lab, is working on amphibians. They're transplanting parts of embryos. Victor Hamburger is forced to flee because he has Jewish ancestry. He ends up at the University of Chicago, where Frank Rattray Lilly is the boss. Lilly says, we don't do amphibians, we do chickens. So if you want to do developmental biology, you need to do chickens. And so Hamburger picks this chicken, Creeper. Creeper is one of the only developmental mutants that's present at the time. So it's one of the only systems where you can do both development and genetics. And what he starts doing is transplanting limb buds. This is very different from the kind of work that was being done with amphibians, and it will actually lead Hamburger's group to discover a nerve growth factor. It becomes incredibly important in the context of World War II when you think about trying to reattach severed limbs. Very important. It's easy to get the bone to reattach, but it's quite hard to get the nerves to reattach. And Hamburger's research with these transplantations in chickens allows some crucial insight in how that can be done. So diversification, definitely. Forced migration forces people into different institutional settings, different material availability that causes them to seek out new kinds of research problems. One other point on this, Spemann hated genetics. He hated geneticists. And so when you were in the Spemann group, even though we know Hamburger had deep interest in genetics from early in his career, he was never allowed to pursue them as a research interest. So leaving Spemann's control also allowed him that intellectual freedom. Now, the reduction question. One of the best sources on biologists who are forced to flee is Uta Deichmann's book, Biologists Under Hitler. Her take on Holtfreter is that there wasn't much change. So even after he was forced to leave Germany, she says he's basically doing the same kind of experiments at both Cambridge and McGill and then at Rochester. And he was already doing sort of a reductionistic research program. And so the sort of implication, sort of was stated and unstated, is that forced migration really had no impact on his research. This is a great book, but I think this is the point at which I'm going to disagree. And what I want to persuade you of today is that, in fact, the forced migration did radically impact Holtfreter, and it is one of the important causes for him to start to move toward lower and lower levels of biological organization. So the forced migration and reductionism in embryology, I think, are closely linked to each other. So did they become more reductionistic? In this case, yes. So the circumstances of his migration contribute to his antipathy to Spemann and to Spamann's holistic approach. And the case we're going to look at in particular is a phenomenon called gastrulation. But before we get to that, let me tell you a little bit more about Holtfreter. Holtfreter was born into a middle-class family in northern Germany. Born in 1901, studies in Rostock in the north and then Leipzig and Freiburg. Gets his PhD in 1924 under Spemann. This is one of, he made many, many sketches. This is actually a scratch sketch. So it's a piece of chalkboard that has black surface coating, and you scratch away at it. So it's a scratch board. And so you can see, Spemann hovering over him as Holtfreter clutches a salamander, desperately trying to come up with a PhD research topic. And this sort of tells you a little bit about their perceived relationship, at least from Holtfreter's position. He really saw Spemann as authoritarian, and that was not incorrect. I mean, this is the way those laboratories were run. Spemann was the absolute authority figure in those laboratories. Nevertheless, Holtfreter was incredibly talented, very, very good with his hands, an incredible experimentalist, and did very, very well. Now, immediately upon graduation in 1924, Holtfreter got a fellowship to go to the Naples Zoological Station in Italy. Fantastic place. This was an amazing place of biological research. Laboratories from all over Europe and the United States would buy a table, buy a research bench, basically, at the zoological station. So Holtfreter goes down there, and he immediately leaves Naples on a boat to go to Ischia. Ischia is a nice little island right off the coast of Naples. Now it's a major resort area. And so for the 2 years that he's basically on fellowship to do research at Naples, he's over on Ischia. And what's he doing? He's painting. He moves to a little town called San Angelo. And San Angelo's church is the church of St. Michael, the Archangel. And so he paints a giant portrait of St. Michael for the church. So that's what he does for 2 years. So the islanders know him as Painter John, and that's kind of his thing, right? So he kind of takes the scientific research money and just escapes from it all, okay? In 1926, he has used up all his money. He's in fact used all of his money altogether. All he has left is enough money for the train ticket to take him back home. He has to have his father pay for the taxi to even get him from the train station back to home. Of course, Spemann hears about all this. He hears that the fact that, Holtfreter isn't at the lab. In fact, Holtfreter's out doing who knows what. And he decides that Holtfreter is not serious and that Holtfreter has been incredibly disrespectful. And he starts a campaign to make sure that Holtfreter will never have employment as a biologist in Germany. So he writes basically to all of the major departments saying, I think it'd be a serious mistake to hire Holtfreter. He's just not serious, right? So Holtfreter is kind of in a bind. He has to figure out what he's going to do for the rest of his life. And he ends up having a choice. So he retrains actually to teach high school science, and he just realizes that's going to be dismal. He takes some of his paintings and drawings to an art representative. So maybe he can make a living as an artist, and they're just not quite good enough, not quite the style that would allow you to support yourself. And so the only job offer that he can get in 1926-27 is on this rock in the middle of the North Sea, Helgoland, okay? So this is a little postcard from Helgoland. You notice there's maybe, 60 houses on this little rock. The major industry is oyster farming, and they desperately need a biologist to help them with oyster culture, hopefully an experimental embryologist type of biologist. The last oyster biologist died of alcohol poisoning. So that sort of gives you an idea of what life is like on Helgoland. But really, it's Holtfreter's only option, right? His bags are packed. He sort of resigned himself. He's going to be the oyster guy on Helgoland for the rest of his life. And he gets a letter. He gets a letter from Otto Mangold. Otto Mangold had been Spamann's star student. And in fact, Mangold had taken Spamann's place at the Kaiser Wilhelm Institutes for Biology in Berlin-Dahlem. And so this grand building with all of its greenhouses and labs is where Mangold is. And Mangold sends Spemann a letter, says, I hear you're looking for work. Would you like to come interview for a position here as a researcher? And so Holtfreter leaves his bags by the door, jumps on the train, and gets to Berlin. He's sitting there in the room with Mangold. And Mangold, they have the interview. And Mangold says, so what do you think basically about this job? As he's doing that, Mangold's secretary brings in the mail, right? And on the top of the stack of mail is a letter from Spemann. And so Mangold says, oh, look, here's a letter from Spemann. We must read it. And so he opens the letter, and the letter instructs Mangold to not hire Holtfreter. It says, whatever you do, don't hire him. Helgoland is where he belongs. So Mangold, even though he's going to turn out to be a Nazi who gets kicked out of the business, turns out to have at least a shred of decency, and turns to Holtfreter and says, well, what would you prefer, Holtfreter? Would you prefer to go to Helgoland and do the oysters? Or would you prefer to come here and be a research associate? And Holtfreter immediately says, please, let me come to Berlin. I'll stop, my errant ways. I will, just not do art anymore. I will just focus in and just do research biology. And so Mangold defies Spemann, hires Holtfreter. Holtfreter actually lives in the attic of the Kaiser Wilhelm Institute. There's a couple other research associates that do it. It's a way that's showing that you're serious. You literally never left that poor building, okay? So he comes to Berlin and he engages in one of the most productive periods of his life, scientifically. So at the lab, one of the things that they're having trouble with is that when you start to culture these embryos, the embryos literally will mold and die. And so in 1931, he develops a solution that's now called Holtfreter solution. It's a modified saline solution that allows the embryos to live. And this sort of contributes to his growing reputation as a wonder in the laboratory. He can do things to these embryos. He can get them to live longer. He can manipulate them with more precision than anybody else in the world. The joke in the laboratory is that his secret is cigarette ash, because he constantly has a cigarette hanging out of his mouth, and they think that he's tipping the cigarette ash into the embryos, and that's what's causing them to live. In fact, he's just incredibly, incredibly good with his hands. In 1931, he makes probably one of his biggest sort of career discoveries, which is that after a talk by Spemann, Holtfreter starts to experiment with the chemical nature of the organizer. So he takes that tissue, and instead of transplanting live tissue from one embryo to another, he actually cooks it. So he takes that little chunk of tissue, cooks it in a flame, and then puts it into the embryo. And surprisingly, the dead tissue causes the embryo to organize. And what that tells Holtfreter is that it's not a property of the living tissue. Instead, it's a chemical, right? So the tissue's dead, but the chemicals are retained. And so there must be a chemical agent that's actually inducing this incredibly complex problem of development. Spemann hates this perspective. He says a dead organizer is a contradiction in itself. For him, he thinks it's a property of the living whole. It's a property of the living tissue that allows it to organize in this magnificent way. And so heat killing a chunk of it is number one, a violation. It's a violence against the organism. And it just doesn't make sense. Nevertheless, you notice that Spemann gets his name on this paper, right? He knows that this is a huge result. And so he sort of insists on sort of being put in the middle of it. During this time period, Holtfreter is sort of involved in all sorts of different activities. The actual photograph of him here is in Richard Goldschmidt's laboratory, which is also in the KWI, and he's sorting Lymantria caterpillars by color. Now, at the end of this period, Holtfreter gets another amazing opportunity. The Rockefeller International Education Board is allowing people to travel to the United States. And so they realize, they get advice that that Holtfreter is one of the premier embryologists in Germany. And the Rockefeller basically sends him over to the United States. The person who he's going to work with is Ross Harrison at Yale. And Harrison and Spemann are best friends. They go way back, and they're constantly exchanging ideas. Basically, Harrison is the best embryologist in the United States, and Spemann is the best embryologist in Germany. And they're working on similar kinds of research. Harrison's probably best known for his work on tissue culture, isolation of different fragments and keeping them alive outside of the organism. This is a photograph of the two of them, so it's Spemann and Harrison. In the lab, this was called the Max and Moritz Photo. So if you're not sort of an aficionado of violent German children's books, Max and Moritz were a horrible set of children, but there were all these children's books where they would basically, cut off their fingers, cut off other people's fingers, do horrible things to each other. So they're basically a rotten pair of children. And so in the lab, that's what they called this photograph. The Spemann-Harrison photograph was the Max and Moritz picture. So Holtfreter gets sent to Harrison. He's going to go to New Haven, and he's going to learn how to do tissue culture. The problem is Holtfreter doesn't need to go to New Haven to learn how to do tissue culture. He's the guy who invented Holtfreter's solution, and he's been doing this kind of tissue culture in Germany quite successfully. And so he doesn't like this idea. He doesn't like the idea that Spemann is once again directing his career. When he gets to the United States, he just basically doesn't come to the lab anymore. He spends an immense amount of time in New York City, which is quite close to New Haven, and really starts going back into art. He paints some beautiful portraits of African-American men and boys, and generally is just not present. And he's not very responsive to Harrison's pleas, to please come to the laboratory, get some work done. And so eventually Harrison just says, you know what, forget it. Just go do whatever you're going to do. I don't want to be responsible. I'm tired of worrying about you. And so quite interestingly, that's what Holtfreter does. He literally just sort of leaves. He's heard that the Grand Canyon is an amazing place to visit, and that one really shouldn't leave the United States without having seen the Grand Canyon. And so he buys a train ticket and he goes out to the Grand Canyon. And you know what? It's amazing. You all should go. And so, once he's at the Grand Canyon, he also hears that Hollywood is a really amazing place to see. And it's not that far. And so he buys another train ticket and ends up in LA. And in LA, he meets some people. One of the things to know about Holtfreter is he liked going to the dirtiest dive bars you could go to. This is not a joke. This is a characteristic of his. He's always going to the roughest parts of town. And so he's in this bar that a lot of sailors go to, and he gets to talking to them, and they say, you know what? You can work your way across the South Pacific on a merchant marine ship, right? So you get free sort of passage by just being willing to do a little bit of work, and it's actually not that hard. And so Holtfreter's heard amazing things about Hawaii, and he decides he's going to just ship out on a merchant marine ship to do a little work, and that's what he does. So he gets to Hawaii. Hawaii's great. He decides he's going to teach himself how to surf, and that doesn't go so well. He actually takes a bad fall and breaks his leg, and so now, there's not much to do. And so he decides to ship out on another merchant marine ship. This time he ends up in Bali, and he loves Bali. Bali is his place. So in Bali, he just sort of immerses himself in the local culture. This is a scratch drawing that he made of Balinese dancers. And the art from the Bali period is quite stunning. It was probably the happiest he's ever been. He loves it. He stays there for over a year, just sort of living in a little Balinese village. No science involved in any of this. 1937 comes, though, and his money has run out. The Rockefeller doesn't even know that he's in Bali, spending all their money there. And he realizes he has to get back on a ship and head back to Germany. Unfortunately for him, Germany had changed quite a bit. And so he comes back to Germany in a period of very strong National Socialism. The Nazi Party has taken power in 1933, and they are really ramping up their program of state control, especially state control of the universities. Holtfreter comes back to a position at the University of Munich. It's not a full professorship, which would have given him a lot of autonomy and control, but it's a better position than he had at the Kaiser Wilhelm Institutes in Berlin. In this period, he's really working on what's called the explant method. So instead of sort of transplanting that embryonic tissue back into an embryo, he's growing it in cell culture, in a petri dish, basically. And what he's doing is he's literally taking apart these embryos piece by piece. He will dissect a tiny, tiny little embryo, like 2 millimeters apart, into over 100, almost 1,000 separate areas, and then grow each of them individually in these watch glasses. What he's trying to figure out is, do these tissues have the ability to develop autonomously? In other words, if you keep them in isolation from everything else, do they have the ability to determine their cell fate? So if you take a little chunk out, will it become nerve cells? Will it specify into nerve cells? Will it become muscle cells or something else? And so this is the kind of question that he's testing. He's trying to figure out where that organization or that determination comes from. Does it come from something that's internal to the cells, or is it coming from some property of the organism as a whole? An amazing set of experiments, right? Incredibly, incredibly detailed and time-consuming, and technically difficult to do these things. In 1938, he is arrested and then dismissed from his position at the University of Munich. This is his actual dismissal letter from the German government. One of the big mysteries is why he was dismissed. There's no official reason given. When I sort of started asking around about this, I went to some of his colleagues, people that had known him quite well. And I said, so, why was Holtfreter forced to leave? Right? It's a political refugee. It's not because he was Jewish. And the answer that I got initially was he was forced to leave because he was attracted to men, right? He's homosexual. And that's it. It was an interesting suggestion, but always hard to verify that, right? So is that just an ugly rumor that's being spread around, or is that the actual reason? It's incredibly plausible, right? One of the things that the Nazis do when they seize power is they strengthen paragraph 175, which is the paragraph, the law, that makes homosexuality among men illegal. So it becomes much stricter. It becomes very easy to condemn someone on the basis of violating paragraph 175, almost to the point of being a joke, right? So if you want to get rid of a noisy neighbor, you just say that they're gay, and the Gestapo will come and take care of them for you. And so some of Holtfreter's colleagues see this as a trumped up charge, right? But it's also not something that they ever write about in print. So it's very hard to get written evidence about this particular reason. So back in the 90s, when I started working on this, I went out and I talked with Hans Holtfreter's widow, Hiroko Holtfreter. Hiroko had been a graduate student of Hans's at the University of Rochester, much younger, married him, and for all intents and purposes, they had a very happy life together in Rochester, New York. I went out there because there was a rumor that none of his letters had survived, that they had all been destroyed when he was forced to flee Nazi Germany. And I was just very interested if there was actually any record of him left. And there was a ton of material in the basement of their house, and she showed it to me, and we had a very nice conversation, but she wouldn't let me actually go through it. Part of that conversation was a conversation about why he was forced to flee Nazi Germany. And so I just asked her, like, do you know why he was had to leave? And she said, well, no, that was before my time. I didn't meet him until much later. And I, sort of very delicately tried to ask her, like, well, there's this rumor that, in fact, Hans was attracted to men, and this was the reason he was denounced. And she sort of paused, and she said, this is what I'm going to say about that. Hans was the most emotionally honest person I've ever met in my entire life. So if you were walking down the street with him and he saw a beautiful boy, he would say, look at that beautiful boy. And that's all she said, right? And so she's just sort of left it there. And I did not want to push on this particular issue. So we left it at that. I knew that if we could persuade her to donate the papers, the books, the movies, all of the material that she had in the house, that this could be a project worth doing. So I talked to her about it. Garland Allen, also a historian of biology, talked to her about it. And Gar got the papers donated to the Marine Biological Laboratory at Woods Hole. This was in around 2000. The MBL then did not process the papers. And one of the things archivists sometimes do is they will not allow anyone to look at the papers until they're processed. And so for almost 18 years, I knew that there were boxes and boxes of papers in the archive at Woods Hole, but I was not allowed to see them. And so it was only 3 years ago that they got a new head of the library and they allowed me to go in and see the papers. And that's where this document comes from. The papers are amazing. They go all the way back to the beginning of his career. So the rumors that there was no record left were wrong. There's literally hour by hour laboratory notebooks of him in the Spemann lab contributing to the kind of work that will win Spemann the Nobel Prize. So it's an extraordinary record. However, there's really no evidence about why he was forced to leave Nazi Germany. There's letters like this, but you notice there's no reason given in this letter. Nevertheless, there are some clues from other sources. So when colleagues of Holtfreter's write to him, other people, he writes this sort of standard letter here on the left, and it's very kind of a vanilla letter. I've been lost my position, it's my fault, I'm writing children's stories, you know, and painting, right? And his friends know him well enough to know that this is weird, like something odd is going on. They think that he's writing this letter under duress, and so they sort of use back channels to figure out what's happened, and sure enough, they find out that he's been arrested by the Gestapo and he's in jail, right? And actually in Joseph Needham's letter from Cambridge, they say he's in a concentration camp and that they have to get him out. And so they literally at the bottom of Needham's letter, he says, we cannot let Holtfreter just get liquidated, right? So they know exactly what's going on. Joseph Needham is a very left-leaning embryologist. He's part of the sort of invisible college at Cambridge. And he knows that he can't go to Berlin because his politics will be known and he'll be suspect immediately. But they try to get one of the Swedish researchers to get down there and kind of figure out what's happening. Eventually, Needham is going to be able to pull Holtfreter out of Munich and bring him to Cambridge. But still, this doesn't really shed light on why he had to flee. We just know that Needham was instrumental in getting him out of Nazi Germany. So after meeting with Hiroko, a couple weeks later, she sent me a letter, and she says, I found the reason Hans had to flee. It turns out that, of course, I wasn't the first person to ask this question, and somebody had asked Hans this question, and he had wrote them a letter. And Hiroko remembered it because she had typed it for him. So here's the story that Holtfreter tells about why he was forced to flee Nazi Germany. As soon as he had gotten back from Bali, 1937, Paris is having an amazing world exposition. And one of the sort of startling features that's captured on this postcard is that sort of on the main route of the exposition, they had the Nazi Pavilion, which is that giant building on the left with the big Nazi eagle on top, opposite the Soviet Pavilion, which sort of has the workers with their torch and hammer and sickle. And so it sort of literally shows you the opposition of right and left right there on the exposition plaza. Part of the exposition is a giant scientific congress. And Holtfreter gets invited and he loves Paris, so he goes. What he doesn't realize is that he's the only German scientist that has agreed to go to this exposition. And that's not necessarily a bad thing, but it puts him in a very different position. As the only German at the conference, he is now the official representative of the German state. If anybody in the Nazi government had known Holtfreter, they would have known not to make him the official representative of the Nazi state. That is a bad idea. So as the sort of German envoy to this meeting, he is told he can only speak in German, right, because he's officially representing Germany. And the honorarium that he gets from this talk must be returned to the German state, right, because he's there on state business. And so Holtfreter gets to Paris, and he's hanging out with his old friends, and his old friends say, Hans, we know that you speak French fluently. Please don't give your talk in German. No one wants to listen to German. Give your talk in French. And so he gives his talk in French, right? So strike one, not good. In the audience, there are clearly people there to keep track of Holtfreter. So there's a person who approaches him who says he's a journalist, But strangely, this journalist always shows up wherever Holtfreter is. Even when Holtfreter goes to a seedy bar in the Algerian section of Paris, the reporter is there. And Hans doesn't like this. And so what he does is in the Algerian bar, he arranges for two beautiful female prostitutes to go and sort of embarrass this journalist slash spy and sort of humiliate him. And it works, and he leaves the bar quite quickly. That's strike number 2. Strike number 3 is that Holtfreter had a really good time in the Algerian district, and he decides that what he needs to do after the conference is go to Algeria, right? And he's got all this money that the Parisians have just given him. And so instead of taking that honorarium back and giving it to the Nazi state, he uses it to have a wonderful time in Algeria. He goes down to Algeria, takes a little trip through the Atlas Mountains, comes back up through Tripoli, and takes the train up through Italy all the way back to Berlin, or Munich at the time. Spends every cent of the money. And so for him, that's his third strike. So for him, the reason that the Gestapo become keenly interested in him is the behavior at this 1937 conference. Now notice the two reasons may not be mutually exclusive. Certainly all three of those things at the conference would have tipped off the German government that he was not playing the way they wanted him to play. And it could also be that he was deposed under paragraph 175. That would have been a very easy pretext for picking him up and arresting him in the German state. So we know that by 1938 he's in jail, desperate to get out, and he ends up going to Cambridge. Cambridge is a great place for him to go. Both Joseph Needham and Hal Waddington had both been to Berlin to work with Mangold and even down to Freiburg to work with Spemann, and they were trying desperately to start a program of experimental embryology in England that would mirror exactly the kinds of experimental embryology research that was being done in Germany at the time. They make a very nice home for him. He's very happy. It's a great research environment with very like-minded people, both politically and experimentally. Holtfreter is fairly liberal in his political beliefs, so he has some sort of political antipathy toward the Nazi state as well, although he's not nearly as outspoken as somebody like Waddington or Needham is about those kind of things. Unfortunately, as England gets pulled farther into hostilities with Germany, even England isn't a good place for Holtfreter to be, especially because Holtfreter's classification is as an enemy alien. Remember, he has no Jewish descent, and so he isn't classified the same way as Jewish refugees were. He's basically seen as almost willingly having left Germany, right? And so he's suspected of being a spy. So one morning, Waddington comes into the laboratory and he finds this written on the lab notebooks. Dear Wad, all aliens are summoned this morning by police. Please look after my things, H, right? Holtfreter was given basically 30 minutes to get down to the police station. All enemy aliens were being arrested and shipped to Canada to be held in a prison camp. Once again, because Holtfreter's status was unclear. It was unclear why he had to leave Nazi Germany, at least from the English perspective. They thought very well he could be sort of a fifth column hiding out in England. And notice this little thing, please look after my things. This is where the rumor starts that all of his things have been destroyed because Waddington did a terrible job looking after his things. But in the letters, we actually are able to find out what he did a terrible job with. He basically mislaid a bunch of sweaters, a raincoat, and some silverware. Okay, so that's what went missing. All of the papers were fine, but the fact that Waddington lost his raincoat really stuck with Holtfreter for a long time. He just thought that Waddington should have been a lot more careful. So Holtfreter ends up in a Canadian prison camp. And in that Canadian prison camp, he's incredibly isolated, very unhappy. What he does is he literally takes the cedar shingles off of the cabin that he's in and paints pictures of Bali. Now, McGill University in Montreal is headed by a biologist named Barrell. And Barrell, along with a bunch of other academics at McGill, realized that in fact a lot of the enemy aliens over there in that camp in Sherbrooke are academics. And one way to really improve McGill's reputation very quickly is just to pull them all out of the concentration camp and hire them at McGill as professors. And so that's what he does. Miguel hires Holtfreter as a professor, brings him in to start doing biological research again. And so even during the Second World War, 41 to 43, Holtfreter is now at McGill and coming back to problems of embryology. And the problem he's coming back to is the problem of gastrulation. How to explain what he calls the harmonious cooperation and orderly pattern of embryonic organization. And this sort of problem had been laid out very crisply in Spamann's 1938 book, which Spemann treated as sort of the culmination of his life's work. What's gastrulation? I want to impress on you sort of the phenomenology of gastrulation. Gastrulation occurs when the embryos have reached a ball of cells stage, and they're going to undergo an amazing transformation where there's basically going to be invagination, the production of a bodily symmetry, an inside and an outside. The Lewis Wolpert jokes that gastrulation is the most important point of your life because it's where you get an inside and an outside, a top and a bottom, a front and a back. And so I want to show you this film. This film was made by John van Eycken in 2018. It's called Becoming. And it's an amphibian embryo going through gastrulation, but it's done under natural light. So it's not totally different from what you would actually see if you were watching this process occur. So just watch. So all these cells are sort of in motion. So this is sort of the end of the process. The next process is going to be called neuralation, which is really the beginning of the nervous system. So you see what's happened is we've gone from a ball. We've had this massive flow of the cells inside of the organism. And then this little groove is actually the protoneural tube. So that's literally sort of the backbone of this amphibian. It now has a head region, a tail region, a dorsal side, a ventral side. It has all of its orientation. And what we've been watching is literally the coordinated motion of thousands and thousands of cells. So this is the problem of gastrulation. How do you explain that, right? That looks massively complicated, controlled, and coordinated, okay? Zip past that. The way that they would have been studying this up to this point is they would sort of be able to watch it literally happen. But then a guy named Walter Vogt, who was a friend of Spamann's, invented this very nice technique of basically what's called vital staining or vital dyeing. He would color coat little agar chips and then kind of paint the surface of the gastrula. And then you would watch the different colors migrate. And so by following the pattern of colors, you'd be able to see where those actual cells end up in the process. And so this is one of the diagrams that he would have done. You can see the sort of red and blue stripes. And then you can literally follow those patterns as gastrulation occurs in order to try to get a handle on what cells are moving where do they end up, all of that kind of thing. So this would have been the sort of the technique that was used to track out those patterns of gastrulation. Now, right before Holtfreter leaves, right until 1938 when he's getting arrested, he's doing those isolation experiments. And he writes this paper about tissue affinity and morphogenesis. So it's about the problem of gastrulation. And look at the way he describes it. So this paper actually gets printed in German by the time he gets to Cambridge. And he describes this process as a systematic organizing process that's recapitulated every time you have development. He says, It's materialized by specialized agencies that come into action successively, that bring around new kinds of arrangements, right? It's like the construction of a building. But the agencies obey their own rules, despite interdependence and common subordination to a building plan. He says it's not about the characterization of the laborers, but the specifications pertaining to work, the requirements, the rules they follow that call out for immediate attention. So the metaphor is that of building a building, right? But we shouldn't be paying attention to the laborers. We should be paying attention to the plan, to the rules by which this coordination takes place. So that's Holtfreter in 1939. Oh, there we go. More specifically, he says there's a problem of this sort of invagination, the movement. He says the most likely factors are mechanical counterpressure from the membranes from osmosis, or a tendency to invaginate. And he says in comparison with physiologically transmitted stimuli, the crude forces of push and pull are of very minor importance. The specific morphogenic tendency here is the true organizing principle. He says we're still far from grasping this as a physiochemical process that's basically described as self-mobilization, right? So what's important here is that he's still accepting this idea that there's an organizing principle. He's still describing it as self-mobilization. He knows that there has to be a mechanical process here, but he's not, he literally says this is of minor importance in trying to understand this problem. By the time he gets to McGill, especially in 42, his opinion has changed. And he writes two very big papers called The Studies of the Mechanics of Gastrulation. And in these papers, he takes a very, very different perspective. It's only been 3 years, but he's had a very big change in the way he will describe this problem. He says the most astonishing integrative character of gastrulation as a whole has kept people like Vogt and Spemann wondering if there's a superior force supervising and directing single part movements. So basically, he's ascribing to Spemann this idea of a controlling force, a controlling organizer, maybe. Spemann, in his book, approved of Vogt's analysis as not a wandering of cells, but passive obedience to a superior force. Okay? If that sounds political, it might be. For Spemann, in his book, he says the sort of motions of gastrulation are ordered according to a definite plan in the sense of von Uexküll. Now, if you haven't spent a time reading von Uexküll, I can't really recommend it. Incredibly dense, sort of theoretical philosopher of biology, theoretical biologist. But von Uexküll's idea of conformity with plan, the idea that Spemann likes, is sort of captured by this semi-intelligible sentence. Once we've accustomed ourselves to the idea that the whole direction of the organism, in the species, in the community, as well as the individual, lies in the hands of supermechanical natural powers, which is to be recognized not only through rules, but acts according to rules, all life on the globe derives new content, which is accessible to investigation. So in other words, the object of investigation here are the rules. It's the plan of this supernatural, supermechanical natural power. Okay, so this sort of captures Spamann's point of view. Holtfreter reacts very strongly against this. He says, look, here we have a mysterious looking case where an organismic principle, which can't be explained by the combination of the parts, seems to pervade the system and exert dictatorial power, right? So this is right in the middle of World War II. Vitalist skeptics, and here he means Spemann, will strongly doubt that further dissecting analysis of a single process will lead to a deeper insight into the inherently superimposed principle of the whole. So what he's ascribing to Spemann is not only a sort of a holism, but an almost supernatural holism, and a holism that agrees with a metaphor of dictatorial power and politics. Now, in cell biology, metaphors to politics are actually quite common. So there's metaphors of cooperation, competition, control. And here what you see is Holtfreter making this, I think, directly, strongly political language, right? He did not need to call it a dictatorial power. And in this context, I think that's quite important. So what's Holtfreter's approach? He says, look, the directed motions here are traced back to single cells, right? So that's the sort of reductionism. The unitary character of their combined effort is based on their arrangement and their local kinetic behavior. He says all of that controlling, that idea of supercellular forces, can be localized and defined physiochemically. So when he's describing mechanics, it's a local mechanics. Each cell is the entity of mechanical action. And so there's no sort of supercellular force. There's definitely no supernatural force acting here. So what does he mean by local cell action? The key cells here are what are called bottle cells. And so what Holtfreter does is he starts to dissect away the cells at exactly that point where the evagination occurs. And you can see these cells labeled B in the diagram. Those are the bottle cells. He will literally dissect those out of these gastrula and watch them act independently. And they have this tendency for elongation. They literally will pull from an anchor point in one direction. So if the bottle cells are pulling inward, and then there's a very tough membrane, it's actually a viscous slime layer around the cell, then what that does is it creates surface tension that pulls in the other cells. So not all of the cells have a tendency to motion, just the bottle cells. The bottle cells pull down, and then the surface tension created by that coat layer pulls in the other cells behind it. So this is sort of the initial instigator of mechanical action in that process of gastrulation. These are images from Holtfreter's notebooks as he's doing these different kinds of experiments and looking at the interaction of these different kinds of physical forces in the embryos. So how do we interpret this claim? The claim about dictatorial power and a superimposed principle of the whole. I want to propose there's two ways to think about this. Holtfreter could be rejecting Spamann's holism on purely scientific grounds, or Holtfreter could be understanding this holism as part of Spamann's sympathy with Nazi ideology. Now, Spemann is at Freiburg. One of Spamann's best friends at Freiburg is this guy named Martin Heidegger. Heidegger had been rector the year before Spemann is rector. And both Heidegger and Spemann are sympathetic to National Socialism, but actually not full-blown National Socialists. They actually think that National Socialism is going to be a good thing for the universities. When Spemann is rector, he actually writes a very famous article about science in the service of the nation. And so interpreting this document becomes the key to figuring out whether or not you really think that Spemann was a Nazi or not. Interestingly, just sort of as an aside about the Heidegger-Spemann connection, the two of them were very close. And so if you were part of Spamann's group, you had to go sit in on Heidegger's lectures. And Heidegger actually ended up writing a set of lectures about embryology where he calls Spemann the ideal philosopher-scientist. And so there's a lot more crosstalk between these two groups. And we actually have some of the Spemann group's notebooks from sitting in on these lectures with Heidegger. And so there's a sort of a strong sort of intellectual and political connection between Heidegger and Spemann with this, it's what I would call sort of an early acceptance of Nazism. Both of them are going to get moved out of the University of Freiburg as you get a process of Nazi consolidation of academia. But the key part there is that he's initially sympathetic. Now, it doesn't really matter what we think about Spamann's Nazism. What matters is what Holtfreter felt about Spamann's Nazism. And there's some evidence that he definitely was very critical of Spamann's attitude, political attitudes, as well as scientific attitudes. So let's look at the first reason. Was he rejecting this based on scientific grounds? In Victor Hamburger's papers, also at the MBL, we have a book manuscript written by Holtfreter. And basically the book is a screed. It's an attack on Spemann. And his assessment of Spemann is that the mysteries of the organizer led Spemann into mysticism. Spemann was awed by unfathomable wisdom of the embryo that suggests to him that the embryo is pervaded by an intrinsic intelligence, which above and beyond physical and chemical processes is the real controller and prime mover of development. Now, the only reason you haven't seen this book is that Victor Hamburger, who had been Spamann's primary assistant, persuaded Holtfreter not to write it. Hamburger was much more sympathetic to Spemann, held him in much higher esteem. Remember, Spemann had tried to kill Holtfreter's career and almost did it. So Hamburger becomes Spemann's defender and actually prevents Holtfreter from writing this attack. By this time, Spemann had also passed away, and there's some evidence that Hamburger thought that this was a very disrespectful thing to do. Clearly wasn't Hamburger's or Holtfreter's idea. So on one ground, Holtfreter just thinks that Spemann is unscientific, right? Appealing to intrinsic intelligences, to things that are not material processes, just seems to him to be mysticism, not science. So on one hand, there's that argument. On the other one, there's this sort of political metaphor. And there's some indication that this was also driving Holtfreter. And we know, for instance, that from letters also in the Hamburger file, Hamburger wrote a defense of Spemann's politics and biology. And in response to that, Hans Misslin, who was a Swiss biologist, said, look, I experienced Spemann in the late Nazi period. Rudi Geige and I went several times to his lectures in Freiburg. And in his last lectures, he found it necessary to compare and identify the unfortunate organizer with the Führer principle. In other words, to make that dictatorial comparison explicit. So instead of talking about the sort of the organizer, he was talking about the Führer, right? The dictator of the cell that controlled the complete process of organization. So as Holtfreter hears these kinds of things, it sort of reinforces in him that not only is Spemann making a sort of weird metaphysical mysticism, but Spemann is also making this political connection that to him is both unfathomable and reprehensible. So to sort of end up here, did Holtfreter's forced migration influence his science? This is a nice Holtfreter diagram. It's called The Only Genius Among Him. And you notice the one duck turning his back to Holtfreter as he lectures them all. The answer is yes. Yes, his forced migration did influence his science, but not in the sort of simple way that Ute Deichmann has argued, right? There's no simple continuation of work from 1939 to 1943. Instead, what we have is a massive break with Spemann's holism. But that break isn't just about sort of wholes and parts. It's also about mechanisms and the appeal to strictly mechanistic or physicochemical processes versus processes that might have been more supernatural. So on the one hand, you get this idea that Spemann was engaged in mysticism from Holtfreter and that good science shouldn't do that. On the other hand, you also had this political dimension to it, right? Holtfreter had been in a prison camp. He felt, once again, very unjustly treated by the Nazi state. And he also felt unjustly treated by Spemann. And the fact that their political ideologies agreed with each other sort of was mutually reinforcing. So there's sort of two senses of the politics of embryology here. There's sort of little p politics about the relationship between Spemann and Holtfreter, which leads him to identify the organizer as a political system. And so moving to this reductionist level of individuals acting autonomously, but with influence, makes more sense to Holtfreter. It agrees with his political vision more. But there's also this big P sense of politics, that in fact there's larger national and international trends in politics that are also relevant to this decision to push embryology toward the individual cellular and even subcellular levels. So that's what I've got. I'd love to hear your questions. Thank you.
Thank you, Mike. Let me remind you how we're going to be proceeding for the Q&A. If you look at the bottom of your screen, you have a small button, Q&A, and you can indicate that you want to ask a question now, actually, by writing your name there. And I will be promoting you to the panelist status, where you can ask your question directly to Mike. And then I will move you back to the attendee status. So the Q&A session is open. Please, if you can, write your name in the Q&A box. All right. While people are deciding what they want to ask, let me ask you a quick question about the timeline. I got somewhat confused at some point. There was so much information I didn't know about some of the timelines in the 1930s about Holtfreter's life. So he goes to Paris for the international exhibition, right? Where he is the only German scientist, and as a result, speaks in the name of Germany and behaves exactly the way he should not do. And then goes to Algeria, then goes back to Germany, is arrested, sent to a camp, and then goes to the UK. That's the order, right? It's first Paris, then his trip to Algeria, then back to Munich, then arrested, then so I mean the obvious question, if that's the right timeline, could he just have been arrested because of his behavior during the Paris International Exhibition? Could that just have been a reason for him to just be demoted from his position and sent to a camp?
So the timeline is exactly right, but he gets back to Munich and he's allowed to go back to work. So it's not like he arrives at the train station and they whisk him away to the camp, right? So he's back going to work. And then in the letters, they say they have trumped up charges, right? That's against him, right? And so those could be the paragraph 175 charges. We don't know. I've tried to get into the Gestapo records and they're very hard to get into and to navigate. But there's some other evidence that maybe that was what was going on. So he's allowed to work for a little while. We also know that he knows this is coming, right? He knows that he can't stay in Germany. And so maybe he's also provoking this a little bit. He's made plans actually to return to Bali and become sort of a quasi-medical practitioner. He sees that as maybe one of his only options of getting out of Germany until Needham comes in and scoops him up.
I see. I mean, I'll let Sandy talk and have further question. I'll come back maybe later. It was incredible individual. I mean, it's just fascinating. Sandy, your turn. You can turn your video on.
Hi, Mike. Can you hear me?
Yep.
Okay. That was fascinating. There's so many interesting parts of it. I wanted to ask you to say something more about this language of dictator and Führer, because the language of science does a couple of things, right? It is both expressive and it's communicative, right? And so one wonders how much of the language is expressing a kind of political adherence or expressing a form of speech, the language of the Führer, that's going to be most understandable by the readers. So I wondered, it's interesting because we have to talk about things in some language, right? Scientists have to use some language to describe what's going on. And the language of the culture is what you grab first, right? So I'm curious about how you might distill out the source of using that kind of language as either being an adherence to a particular political view or just reflective of the culture that's adhering to that view.
Yeah, it's actually quite tricky. And so if you go back to the ways Spemann first described the organizer, he literally is using this word Organisator in German. And so that's the word he uses throughout all the way until you get to these lectures in 1939. And then he substitutes in the Führer language, which is quite odd. And that's right after he was rector of Freiburg, where he's actually making comments about the state and science together. And so to people like Mislin, they definitely saw that as trying to please the Nazi hierarchy, right? They saw that as an explicit political appeasement of some sort. And that's probably the way that Spemann would have read that situation as well. I think he already thought that Spemann was much more politically conservative and already a friend of the Nazi state. Remember, Spemann went along with having all of his ex-Jewish students deported, right? So Spemann's happy to see, not super happy, but he raises no objection. He's like, well, to Hamburger says, well, that's what you have to do. You're not German anymore. And so the politics, so what matters for the story is that how did Hamburger view this weird political situation? So I agree the language is ambiguous and it could have been just sort of a very localized instrumental use. But then you see Holtfreter saying, look, he's creating a system of dictatorial control within the cells. And I just don't think that that language is accidental, especially when you compare the 39 articles with the 43 articles. That's a pretty rapid shift from a metaphor of building a building to having a dictator control the organism. So that's kind of, yeah, it's not perfect. We don't always get perfect evidence, but
No.
it's sort of, it's leaning me that way.
Okay, thank you.
Mahi, your turn.
Hi, Mike. Thank you so much for the talk. It was super fascinating. I just had a question sort of about the generalization of the account that you've given to other disciplines. So some of the things that you were saying really reminded me of discussions of things like physiology or like neurobiology and how you have that shift after like from immigrant scientists. I was just wondering like what you thought about that narrative about the move to reductionist, like sort of science in that move, and whether you see the same kind of thing in other parts of biology, but like maybe also physics. Like, does the story look different there?
Yeah, the bigger story about reduction that people are kind of coming against is that, you know, the obvious story is that, well, biology becomes reductionistic when you get molecular biology, right? And so as with the rise of molecular biology in the 40s and 50s, then of course every field becomes more reductionistic as they adopt these molecular concepts, attitudes, methods, and so on. And so for a long time, that was the sort of reigning story, right? And you know, if that's your assumption going in, then you look for molecules, you find molecules, you're confirmed, right? And so part of this is part of stepping back and going, what about before molecular biology? Were people actually thinking in reductionistic terms and maybe not going, so when he kills the organizer, that does start a period called biochemical embryology because it has to be a chemical. Really hard to find that chemical, like takes 30 years to find that chemical. But you know, you sort of see the clues of them going for that and then going to these subcellular and very mechanistic kind of explanations is sort of this pre-molecular kind of reductionism. And, you know, being more historical, I would say, I don't know, I haven't looked through all of those cases, but I would be willing to bet that the old story that it's molecular biology taking the lead here is probably not going to hold for those sciences either. That I bet that there were earlier forms of biochemical work, you know, especially in the neuro, if they're looking at all like calcium ion channels, you know, the effect of calcium in cells. There's all this early colloidal chemistry that basically was biochemistry before there was formal molecular biology. So I would say that reductionistic move is going to get pushed back. It's just, that's the kind of historical moment we're in right now, is actually investigating those earlier pushes toward reductionism.
Yeah, I mean, I think I was also interested in the political connection. So do you see the same kind of move from like German physicists who went to the US, or because there were also like a lot of yeah, there were lots of kind of mysticism trends in some of like the early quantum mechanics work, and I'm just wondering like how that got framed by them. Like, do you think there's a similar like political equivalence in that work?
Yeah, in the literature on exiles, when they talk about the physicists, they never actually have to get into the science that much because the Nazis created Aryan physics, and so you could just sort of stop your story right there, right? If Einstein was doing Jewish physics and other non-Jewish biologists or Germans were doing Aryan physics, then the politics becomes super clear, right? I don't know. I'd have to go back and see if anybody then makes the connection to sort of German mysticism or mystical sort of interpretations of those other sciences as a result of that. That's a good question. You know, there is that book about by the person at Harvard whose name just zipped out of my head. She talks about von Uxkol and the cognitive dimensions. And so she talks quite a bit about that sort of mystical tradition. But remember, the Nazis were, they would adopt anything, right? And they found ways to make all different kind of scientific positions agree with their ideology. So that there's not sort of one single answer that they're all, and they, of course, they weren't consistent either.
Thank you.
Thanks.
Thank you, Eric Anderson. So you can unmute yourself and turn the camera on to ask your question. All right, thank you very much.
Hi, Mike. Thanks. That was really awesome. My question is, I guess it's not a very well-formed question, but I'm very taken with all these artworks that you've shown in your presentation, and you know, they sort of have this clearly very irreverent vision of the world, and you know, the people that he knew. And it kind of got me thinking about, you know, the artworks of Ernst Haeckel from the previous century, and how different they were, and how they sort of presented this, you know, almost kind of transcendent order and symmetry of nature and all this kind of thing. And I'm just wondering if you've thought at all about how Holtfreter's artwork influenced people's perception of him in, you know, throughout his life, and in these troubles that he faced with the Nazis and things like that. Does it reflect in any way, you know, a difference in his kind of vision of nature or of science? You know, any kind of things you've thought about in with respect to that?
Yeah, if you look at like the styles of his sketches, it very much fits kind of like almost a style of political or satirical cartoon sketching that was very popular in the Weimar period. And so you can definitely see similarities to that. His paintings are much more in a romantic style. They're big oil canvases. The thing about his art is that he always said that it was private. Right? He did it for him. It was like his therapy. And so he would do these amazing sort of scratches. He would do sketches. He would do watercolors. He would do oil paintings. He also did sculpture. He was incredibly good at sculpture, which is not surprising, right? He's good with his hands, period. But they're very private. And so when I visited, for instance, his house in Rochester, Hiroko took me into the garage, and there was a giant piece of plywood, like 8 feet by 5 feet, that was just an incredible Balinese landscape that he had just sat out there in the garage and painted for himself. And so this is really, for me, like an almost internal self-expression. And there's tons of art. I mean, we could have made this twice as long and showed you a picture for every mood that he's in. And so I use it more as like I don't know, sort of a different way of glimpsing into what might be going on in his head. You know, the only person that he sent pictures to was also Hamburger. So every Hamburger letter had like a really funny little sketch in it as well. But then when you look at his scientific notebooks, his artistic ability is on full display. I mean, his ability to sketch those cells and to sketch them very quickly and beautifully is quite stunning. And so I think his ability to render and to capture these complex three-dimensional processes, there's no photomicrographs. He does not take a picture of anything, right? He does later take movies, which is quite interesting because he's studying how amoebas move, but that's like his retirement project. And so there's lots of films of that. And within the Spemann group, there were movies. They did take movies of gastrulation, and they would send joke movies back and forth to Harrison. And I'm on the hunt for the joke movies because I think those would be very interesting if they're like sending birthday greetings or they're telling, you know, they're having funny plays. Scientists at this point in time, I don't know if you know like this part about their culture, but they're always putting on shows for each other and inventing stupid songs. And they had sing-along like every Wednesday night at Woods Hole. And so all this kinds of expression is also part of the community culture. It's what kind of binds them together. So there's kind of multi-level things going on here with the art. You know, if I were to back up from this, I'd say the art's super important to his science because it's those abilities literally to represent and to manipulate, that make him this incredible experimentalist.
Thanks.
Thank you. Does anyone else have any question? Yeah. Let me, Greg, I'm going to promote you to panelist. All right. Greg, you can turn your
yes.
mic.
Okay. Thank you. Okay. Can you hear me? Can you see me? I think I just kicked myself out of the room. Hey, very quickly, since two very basic questions. One would be in the late 1930s when they had every reason to sort of kick him out, but it seems as though they waited a while. Was that because of a sort of general interest in Nazi interest in biology, where even if this guy was strange, at least he wasn't Jewish or anything, so maybe we'll sort of let him play around with his stuff for a while to see what he can come up with. And then secondly, after 1953, did he pursue any of his ideas with respect to the double helix, the selective unzippings and everything like that? Or did that not proceed far enough until his retirement and passing?
Yeah, both good questions. So in the late 1930s, he's very well thought of. And it's not clear that the Nazis see him as a valuable scientist, like somebody worth saving. Otto Warburg is a biochemist in Berlin. He's Jewish, and he actually is not forced to flee. And he's not forced to flee because he is measuring rates of respiration. And cancer cells have higher rates of respiration than any other cells. And so Hitler makes an exception for Warburg because he thinks that Warburg is the world's most advanced cancer biologist at the time, and Hitler is afraid that he's going to get cancer, right? And so they are willing to make exceptions. I don't think any of that thinking applied to Holtfreter. He's already kind of politically left. He's definitely got this anti-authoritarian tendency. He's definitely not a good rule follower. So I think there was lots of strikes. I think they were literally trying to manufacture this case and figure out how to do it. The problem is that at this point, they're not actually at war with anyone. And so they actually are still trying to find justifications for what they do. And I think that's partly of what slowed down this process in the late 1930s. Now, what does he think about sort of the molecular turn in the mid-1950s? Even though he leads this reductionist move towards cellular and subcellular, he's an organismal biologist, right, at heart. And I found no evidence that he actually takes on anything molecular. What he does in his later career is he and a graduate student published this incredibly important paper called the Townes-Holtfreter paper, which is about cell affinity, which becomes absolutely crucial to cancer research. And so the idea is that you can disaggregate an organism and their cells will all go back to their correct layers and affinities with each other. So it's once again trying to get at that idea of organization and how do cells understand where they belong in an embryo. And so it's still very experimental, very manipulative. Other people are going to take it in that more biochemical, more molecular tradition. And so people like Hamburger, remember Hamburger goes to genetics. The whole double helix thing fits in much better with Hamburger's way of thinking. And so Hamburger is much more readily picking up the DNA, RNA, protein kind of story and integrating it out within development than somebody like Holtfreter, who's still sort of trying to figure out those processes of cellular mechanics.
Thank you.
Thank you. I think there's no other question, at least for the time being. So, but let me ask one more, and someone will pop up. So I was interested by the fact that there seems to be no evidence about his sexual orientation, about the fact that he might have been gay, might have contributed to his being removed from his post and have to leave Germany. On the other hand, if you look at his lifestyle, it's actually quite characteristic of the lifestyle of men in the 1920s and 1930s, having at least part of their sexual life being gay, including going to Algeria. So Algeria was actually a very traditional destination for European gay men. Of course, starting in the 19th century, Oscar Wilde, of course, traveled to Algeria many times, and as part of his homosexual life. André Gide in France, a famous French writer, of course, went to Algeria and as part of his homosexual life. It was actually a common destination for gay men of the time. Now, it's of course very weak evidence, if evidence at all. But of course, liking the CD bars, and there's, it's not, of course, the only man of the 1930s with having a gay life on the side, and indeed going to CD bars and to a place where matelots and other people go. There's all this very anecdotal evidence on the side that does not clinch the case, but it's actually at the very least suggestive.
I think it's super suggestive, and the fact that people who knew him this would be the first thing they suggested was interesting, right? The portraits that he paints of African-American men in Harlem, right? They're all men. He doesn't paint portraits of women ever. That was kind of interesting. The Algerian connection is very interesting. When he's on Ischia, he goes to San Angelo. San Angelo was a famous gay beach in the 1930s, right? And you ask, Mike, how do you know about gay beaches from the 1930s on Italian islands? I gave this lecture, and Jerry Weissman, who is this old biologist, this is like a week before he died, right after the talk, he called me up and he said, I was in Italy in 1934, and I went to Ischia, but my dad knew I was going to Ischia, and he said, whatever you do, don't go to San Angelo, right? That's the gay beach on Ischia, right? And so he has sort of had firsthand experience, and so it was very telling that that's where Holtfreter goes and spends, an entire year and a half is hanging out on that beach. Now, that's all still very indirect. There's a couple little letters from other people going, could it be true? The rumors that we're hearing for why he was kicked out, but they never actually put the rumors in print, right? When I've gone and I've talked to other sort of historians of sexuality, I'm like, am I going to get better evidence than this? And they said, look, that's all pretty compelling, right? And if you're waiting for the confessional letter, you're never going to find it. This may be as good as it gets. And so that's why I kind of tell the story two ways. I think the Paris incident is definitely important. I think there's probably other incidents. If I could find out that it really was a paragraph 175, that would be another piece of incidental, information that would really sort of seal up the story.
And he had no political orientation that I would say would have put him against the Nazi orientation. He was not a communist sympathizer or socialist sympathizer.
Yeah, he's hanging out with all the weird bohemian artists. And so he's definitely hanging out with that left thing. But he's like not a member of any party. He doesn't spend his weekends at marches or rallies or beer halls or, there's nothing like that, the level of activity that you think would have put him on the radar of the Gestapo.
Yeah, that's fascinating. All right, any other question? And we are reaching anyway the 1:30 time mark. So if we have no other question, thank you, Mike, for this fascinating talk about a fascinating man indeed, and a great scientist. And we'll see all of you on Friday for Mark Wilson's talk on wavelengths. Have a good day. Bye.